Okay, welcome everyone to today's edition of Tissue Talks. Our usual reminder to please post your questions using Q&A feature that you have at the bottom right of your screen and not the chat. And our speaker will answer as many questions as possible at the end of his talk. It's my real pleasure to introduce our speaker today. This is Professor Tal Dvir from Tel Aviv University in Israel. Tal obtained his bachelor's degree in 2003 and his PhD degree 5 years later from the Faculty of Engineering at Ben Gurion University of the Negev. His PhD research was focused on cardiac tissue engineering and regeneration, and this is really an area that he's pursuing very strongly up to today. He then moved to the US for his postdoctoral studies under mentorship of Professor Bob Langer in the Department of Chemical Engineering at that time at MIT. His postdoc research shifted to some extent into design and utilization of advanced materials that find utility in tissue engineering and tissue regeneration, both in vitro and in vivo. In 2011, after his postdoc, Tal was recruited to the Department of Biotechnology at Tel Aviv University, and he was also given directorship of the Center for Nanotechnology at Tel Aviv University. His laboratory is focused on tissue engineering and regenerative medicine. Since 2021, he also has a dual appointment at the Department of Biomedical Engineering at Tel Aviv. Tal's Tal's laboratory is actually over the years focused on designing and developing smart biomaterials and nanomaterials and new technologies for engineering complex tissues and organs that start with the heart, but actually he has also done research in of the brain tissue, spinal cord, intestinal, eye tissues, and more. Among the technologies that he has developed in his lab are new modalities for three-dimensional printing of the tissues and organs, a very unique approach to building in electronics into the engineered tissues, in particular implantable electronics, and in the development of personalized engineered tissue implants. His research really has a major clinical potential, and I believe that he is one of the people who are truly making a change in how the diseases are treated, how the treatments are implemented, and his work may prolong and improve the quality of life as it is being translated. Tal also, not surprisingly, has numerous patents that are protecting technologies that he has developed. He obtained a number of awards, and I will just mention one. This is a major award, which is Tao Book Da Vinci Award for the year of 2023. And it was given to him for his overall contributions to the field of regenerative medicine. So in addition to being at the forefront of our field, I think Tal also has credit for training some of the best minds and most prominent rising stars that we have today in our field. So Tal, thank you so much for being with us today, and you can start sharing your screen, please.
Thank you, Gordana, for the introduction, and let's see that it works. Can you see my presentation?
Yes, just change to presentation mode.
Yeah, can you see it now?
Not yet. Yeah, it's perfect. Thank you.
Okay, so thank you, Gordana, for the nice and long introduction. As Gordana said, my lab at Tel Aviv University is working on tissue engineering, and we're engineering different types of tissues. We've started with the heart and then the brain, spinal cord, intestine, kidneys, retina, and other tissues. And I'll give an overview of what we do in the lab and how we do the tissue engineering and what's our approach. So when we talk about tissue engineering, so most of you know this scheme, but this is the main concept. But what do we do in the lab? So we take cells, and preferably we take the cells from the patient because we want the cells to be compatible. We cultivate the cells in 2D flasks or Petri dishes, and then we need to create tissues. And as you all know, tissues in the body are not just the cells. The cells are very important, but the biomaterial in between the cells is important as well. And what we do in the lab, we develop different types of materials, three-dimensional materials, porous materials, in which we seed the cells. We can add small molecules, nanoparticles, growth factors, or particles slowly releasing factors. And essentially, we're doing everything we can to allow the cells assemble into a functioning tissue, which can then be taken and transplanted instead of the defective tissue. And when we talk about cardiac tissue engineering, and we cannot, of course, take cardiomyocytes from the patient. First, we cannot harvest cells from the heart, and also, as you may know, cardiomyocytes are not proliferating cells, so we do not have enough cells. So what can we do, since stem cells or stem cells in the body cannot differentiate to cardiomyocytes? We need to use either embryonic stem cells, but we do not want to provoke an immune response. So what we use is human-induced pluripotent stem cells. And we can differentiate the cells to different cell types of the heart: fibroblast as a connective tissue, cardiomyocytes, and subtypes of cardiomyocytes, smooth muscle cells, and endothelial cells for the blood vessels. And then we can create the tissues in the lab. We can create 2D constructs that may be useful maybe to screen drugs. Not ideal for that, but they are used to screen drugs. Or we can create more complex tissues, three-dimensional cardiac patches with blood vessels to replace the defective tissue on the left ventricle. Or we can start now by playing and maybe printing or engineering the entire heart. And I'll explain how we do that. So when we talk about the biomaterial that we use, it can be in different forms. We can use hydrogels. In this case, as you can see on the left side, this is a hyaluronic acid hydrogel. We can mix the cells within and either inject these cell-containing hydrogels, or we can transplant them. We can use preformed scaffolds, some kind of sponges with pores. We seed the cells within, and then we can transplant these tissues. Or we can use fibrous scaffolds like these PLGA electrospan fibers that we see on the right side. And these are really resembling the morphology of the extracellular matrix. And when we talk about the heart, initially when we started working on that, we want to evaluate how the extracellular matrix of the heart looks like. And what we've done, we took hearts, removed all the cells, and we were left only with the biomaterial in between the cells, the extracellular matrix. We could see that there are nanofibers, microfibers, aligned fibers, randomly oriented fibers. And initially, we wanted to evaluate what's the role of the different fiber populations. And we created synthetically these fibers, the microfibers, nanofibers, aligned or randomly oriented ones. And I'll give you two examples of how the cells react differently to different scaffolds or different morphologies of the material. So in the case of the parallel fibers compared to the randomly oriented fibers, you can see that the cells are really aligned on the parallel fibers. And this is extremely important when we talk about engineering the heart, because we want to have a nice propagation of the electrical signal from one spot to another. And with the randomly oriented fibers, we cannot achieve that because there are contractions in different places to different directions, and this is not ideal. So this is one example. Another example is the effect of fiber diameter. And here you can see on the left side nanofibers. In the middle, you can see 1 micrometer fibers. And on the right side, you can see 5 micrometer fibers. And when you culture cardiac cells on these fibers, in this case, we cultivated dissociated cells, you can see that the cells react completely differently on the nanofibers, and this is uniquely to cardiomyocytes. Other cells may react differently, but cardiomyocytes don't really like the nanofibers. They prefer the bigger ones. You can see on the left side that they are still rounded. Their sarcomeres are not really developed. And on the right side, on the bigger fibers, you can see elongated cells, pronounced and massive striation, very nice sarcomeres. And this is important when we talk about creating cardiac patches that will contract nicely in the lab. So if we take the morphology of the fibers into account and other things that I haven't talked about, like the mechanical properties of the materials, the biochemical content of the materials, we can create cardiac patches like this. This is can be a few centimeters in diameter, a couple of millimeters in thickness, and they contract nicely in the lab for a long period of time without external stimulation. We can take these patches, transplant them on animals' heart to regenerate, and we can see very nicely that the ejection fraction is elevated when we use such cardiac patches. Now, when we talk about human hearts, we cannot use 2 millimeter thick patches because the human heart wall is about 1 centimeter in thickness. And when we try to engineer such thick scaffolds and engineer such thick tissues, there are mass transfer limitations. Oxygen cannot penetrate deep into the tissue, and the core tissues simply die. And to tackle that, what we've decided to do is what we call a bottom-up approach to assemble thick cardiac patches. And what we've done here, we created many, many thin layers of tissues. So we took electrospan fibers, we patterned them using a laser. For example, we had many, many grooved layers for cardiomyocytes, for cardiac cells. We seeded the cells, they were anisotropic. We had more layers with channels for endothelial cells to create the blood vessels, honeycomb structures to accommodate VEGF releasing particles, and also honeycomb structures to accommodate dexamethasone releasing particles to avoid rejection. So we grew these thin layers separately with enough oxygen, and just prior to transplantation, we assemble them into a thick tissue. And you can see here one example with the release of VEGS and without the release of VEGS, this 1 centimeter thick tissue in vivo. And we can see nice penetration of blood vessels into this tissue, and it is very well nourished. So we have now a way to create thick tissues that are appropriate for a human to regenerate the human heart. But when we talk about the materials that are used, most of the time we're using synthetic materials. And synthetic materials, when we talk about electrogenic tissues, tissues that transfer the electrical signal, once we transplant these synthetic materials, there are fibrotic tissues around them, and these fibrotic layers, they stop the electrical signal. And people thought, well, let's not use synthetic materials. Let's use perhaps materials that come from pigs. And there are millions of people who already received these materials. However, they also receive immunosuppressors to reduce the immune response because there are certain antigens that the pigs have. For example, this sugar, alpha-gal, the pigs have them, but we humans do not have them. Additionally, the ECM proteins that we share with pigs, there are differences in the amino acid sequences. If you look at collagen 4, for example, you can see up to 37% difference in the amino acid sequence, and this means that the protein folds differently, and eventually this provokes an immune response. So what we've decided to do was to create completely autologous materials for autologous tissues. And since we cannot take the heart and create scaffolding material from the heart, we decided to use fatty tissue, the omentum. The omentum is a fatty tissue in the belly, which has remarkable regenerative capabilities. It serves in the body as a depot for stem cells. And what we've done here, we took this tissue out, we isolated the cells, reprogrammed them to become induced pluripotent stem cells. And the ECM of this tissue was processed to become a scaffolding material. Now we can take the cells, seed them in this scaffold, differentiate the cells to the different tissues that we want, and create completely autologous tissues that will not provoke an immune response. And if we look at the material, this is how it looks like. This fibrous structure. Cardiomyocytes really like these materials. They can even remodel the material to have aligned fibers and aligned tissues. We can create blood vessels within. Eventually, these tissues, these autologous tissues, contract nicely in the lab. Now, if we want to evaluate the immune response to the patient-specific materials and compare them to pigs' materials or to allogeneic materials, that meaning that they come from other patients, we can do that by simulating the immune response in a dish. What we do here, we take donors' blood and donors' tissues. We isolate the antigen-presenting cells and the T cells. We incubate the antigen-presenting cells either with pigs' materials or with human materials, either from the same patient or from other donors. And after 2 days, we added T cells. And what we were able to see was actually two nice things. First of all, something that was logical to us, that there is a difference between the pigs' materials and the human materials. But most importantly, we saw that each of us donors react differently to materials that either come from us or from other donors. And this is why we decided that there is a need for really personalized materials as well, not just patient-specific cells. We've shown it also in animals that the autologous materials provoke less immune response. And actually what you see here in red is just the effect of the incision that we created, but you can see that the materials that come from other animals provoke more immune response. So now we have materials or completely autologous tissues that we can transplant. And we do it in animal models of myocardial infarction. We create an open chest surgery and we transplant these patches. And from this movie, you can see two things. First of all, that there's an open chest surgery, which is not ideal for patients who just had the heart attack. And the other thing is, I'll talk about it later, are the two stitches that you can see here that secure these patches or this patch to the heart. So first, to avoid the open chest surgery, what we've decided to do was to take the material that comes from the patient and process it to become liquid in room temperature. Now we can mix the cells here. And in 37 degrees, this material simply solidifies. So now we can inject it wherever we want in the body, and it will basically form the tissue in the body, either in the heart, in the brain, in the spinal cord, wherever we want. So if we look at this approach, we can take the omentum from the patient, extract the stromal cells, reprogram them to become induced pluripotent stem cells. The ECM of the tissue is processed to be a personalized hydrogel. Now we can mix the cells here and we can mimic the embryonic development of different tissues. For example, we can mimic the embryonic development of adipogenic tissues to create adipogenic implants for reconstructive surgeries. We can create motor neuron implants for spinal cord injuries, cortical implants for brain trauma and stroke, dopaminergic implants for Parkinson, and cardiovascular implants for cardiovascular diseases. And we can basically also use more advanced technologies such as 3D printing and microfluidics to create more complex tissues, as I'll show in a few slides. And just to disclose, based on this technology, we formed a few years ago Matriself. Matriself is a matrix and cells and self. So it was formed in 2019, and I'll explain what we do in the company soon. So here you can see these cardiac implants. You can see that they contract, they transfer the electrical signal really nicely. You can also see that once they are injected into the heart, the infarcted heart, we can elevate the ejection fraction in small animals and in large animals. And then we said, well, we have these hydrogels that can solidify in 37 degrees. What else can we do with them? And we decided, it was a long time ago when we started doing 3D printing. I think it was about 2014, 15 when we started working on that. And how do we print the tissues? So we look at CT images of the infarcted heart, as you can see here in this human image. We isolate the blood vessels. These are the big blood vessels. We cannot see in CT the smaller ones, but we transfer these vessels to the computer. And then we need to see whether these blood vessels are enough to nourish the tissue, or we need to add more blood vessels. And this we do by using mathematical modeling. We start with the basic architecture of the network, and then we add more and more blood vessels until the entire patch is completely nourished. And then we move on to really print this tissue in one of the printers in the lab. And we're printing the parenchymal tissue, the cardiomyocytes, and the endothelial cells for the internal structure of the blood vessels, and either smooth muscle cells or fibroblasts for the to cover these blood vessels. And eventually we're getting this cardiac patch with this blood vessel network, which fits the anatomy of the patient, the biochemical properties of the patient, and also the cellular properties of the patient, because everything comes from the patient, from the material to the cells. Now, if we look at the cross-section of one of the blood vessels, you can see the endothelial cells are lining up really nicely on the walls of the vessel. You can see here, in this case, the fibroblasts are really covering these blood vessels. And after 2 weeks, we can also see, this you can see on the right side, that new blood vessels, new capillaries are sprouting out of these big blood vessels. So currently, the limitations of the printers are we can print about 100 micrometer blood vessels in diameter. We cannot print smaller ones, and I'll explain in a few slides what we do when we want to create or to control the structure of the capillaries. So this is the cardiac patch, which contract nicely. You can see here the either the entrance to the inlet of the vessel and the blood vessel itself, and you can see that the tissue contracts nicely. Now we can use these tissues to regenerate the infarcted heart, but we also can use them to do something new. Instead of, for example, screening drugs in organ on a chip or in 2D really thin layers of cardiomyocytes, we can do it by injecting the medium with the drugs into the blood vessels, and then we can evaluate the effect of the drugs, how they go out of the blood vessels and how they affect the tissue. And in this case, we can see how they affect, how the drugs affect engineered tissues. And this was in collaboration with Bayer. Now, I talked about the fact that we want to avoid open chest surgery. And if we print these patches, we need to transplant them. Now, they're big patches. They sometimes can reach to a few centimeters, 5, 6 centimeters in diameter. And if we try to maybe inject these tissues or bring them into the heart with a minimally invasive technique, they simply collapse. And here what we've decided to do is to reinforce the tissue, to cross-link it, but to cross-link it in a clever way only after the tissue is assembled. And here we use small molecules, oxidized sucrose. We wait until the tissue is really matured, the cells or the tissue contracts nicely. We add this molecule, and this molecule really cross-links the tissue, keeps it viable, and then we can inject really large tissues. And here you can see before injection and after injection, you can basically see that the tissue is still functional. You can see it by these heat maps showing that there's a nice propagation of the electrical signal before and after injection. So we thought that we're 3D printing tissues, but then we thought, well, it's not real 3D tissues because they indeed have height, but they're not really volumetric tissues. So we call it in the lab 2.5D printing. And when we want to move on to print more complex or volumetric structures, we need to move on to print in support bath with small nanoparticles. These nanoparticles can hold the space until the tissue is solidified, and then we can remove these particles. And this way, we're able to create really volumetric structures. As you can see here, I really like this movie on the right side. You can see a ball within a ball, and this is from the patient's own materials and the patient's own cells. Now, after we print it and we heat it up and it solidifies, we can add an enzyme to basically break these particles, and they are washed away, and we're left only with the engineered volumetric structure. And this allowed us in 2019 to print the whole hearts with the major blood vessels, with the ventricles or with the four-chamber structures. And the card, you can see it here in these pictures. You can see in the middle of the printing and the end product. And the cells within these hearts contracted, but they did not contract synchronously. We needed to teach them how to talk with each other on the electrical level. And for that, what we've decided to do was to use to mature these hearts in specialized custom-made bioreactors. So here what we do, we basically hang these hearts, we circulate the culture medium through these hearts, through the major blood vessels, and we provide electrical stimulation for maturation of these hearts. It didn't work perfectly, so what we've decided to do next was to use the body as the bioreactor. Since we can create these hearts in any shape that we wish, we closed all the inlets and outlets, just leaving one inlet and one outlet open. And we transplanted these hearts in a tortopic model of in rats. So you can see here the transplantation. This is the engineered heart when we connect it to an artery in the stomach. And after 7 days, we can take these hearts out. They got signals from the body, from the animal. And if you look at the contraction of these hearts, you can see them contracting very nicely in the lab after we extract them from the animal. If you look at the electrical signal, you can see that they transfer the electrical signal really nicely. So this was really nice for us. We were very excited. But in this case, what we had was a mixed population of cardiac cells. We had ventricular cells, we had atrial cells, maybe pacemaker cells. And next, we thought that we want to differentiate the cells to all cardiomyocyte subtypes and place them in the right place in the right location within the printed hearts. So we differentiated the cells to atrial cells, ventricular cells, pacemaker cells, as you can see by these markers and these functions. And initially, before printing the heart, we want to evaluate whether the pacemaker cells can interact with the other cells and really activate them, or whether the ventricular cells or atrial cells will start their own activation and there will not be order within these engineered structures. And we thought of doing that in a simpler model than the heart. So we printed rings. And as you can see here, these are ventricular rings without pacemakers. Each time the signal starts from a different place. There's no control. And when we do it and we print the pacemaker cells in one spot, you can see it here. First of all, it beats stronger and faster. And also, each time it starts from the pacemaker cells area. And this led us to believe that the pacemakers can be integrated with atrial cells and ventricular cells. And we moved on to print the entire heart with the atria, with the ventricles, with the pacemaker cells. And as you can see, these hearts contract nicely. We moved on to do echocardiography and on these hearts, and you can see it here. And maybe you'll appreciate it's still not enough, but the ejection fraction of these hearts is about 16%, which is not above 40 as we wish, but we're on the way. So if you remember, I talked about the smaller blood vessels and the fact that the resolution of the printers cannot allow us to basically create these or to print the small scale capillaries. And what we decided, before that, I'll talk for a minute about the blood vessels large blood vessels that we can print for bypass surgeries. So here we developed a way to print these blood vessels with openings wherever we want to create Y shape or whatever shape that we wish. You can see it here. We can create different layers, for example, for the endothelial cells, smooth muscle cells, fibroblasts. We can create any structure that we wish, and we're now transplanting them in animals to evaluate whether these engineered blood vessels can work with the pressure in the body. So now when we really move on to the small scale capillaries, we cannot create them with the normal printers. And we thought maybe we do not need to change the printer. Maybe we need to change something later on after printing. And we call it 4D printing because what we do here, we print tissue with large blood vessels. And in the areas where we want the blood vessels to become smaller afterwards, we print tiny nanoparticles. And these tiny nanoparticles in 37 degrees, they pull the blood vessels to create a small vessel. So we print them in 100 micrometer in diameter. And then, as you can see, this is how they interact with the fibers, these particles. This is a FIB-SEM SEM image. As you can see here, the particles interact with the fibers of the bioink. And basically, look at this movie and look at these arrows. In 37 degrees, everything shrinks. Not everything. Selective areas within the tissue shrink. And these cardiac patches contract, and they have small-scale blood vessels and the large blood vessels as well. Now, this was about the heart. We'll move on to the spinal cord. We do the same thing with the omentum, with the iPSCs, put them in the hydrogel, mimic the embryonic development of the spinal cord. And after 30 days, you can see there are neuronal networks, 3D neuronal networks within these hydrogels. Now we can take them and transplant them in a chronic model of spinal cord injuries. And here you can see in this MRI images, you can see the nice recovery of the spinal cord using this approach. And you can see with the hydrogel, the untreated or just the cells, there are still cuts or and the spinal cord or the tracts are not really complete. And if we look at the animals behavior, and I remind you, these are chronic, this is chronic model of spinal cord injury, meaning in this animal, it's a few months after the injury was done. Only then we treated these animals, and this is equivalent to more than 1 year in humans after scar tissue is already formed in plateau. And you can see here the untreated animals. You can see that the animal is not walking perfectly. One of the legs is not really operating. And more than 80% of the animals walk like that. And this was really surprising and nice to see. And based on that, we formed the company Matrisolve, and hopefully in a few months we'll be already in, treating humans in Israel. We got approval from the for 8 patients, and it will be done in the really next really soon. This is some usability studies that we've done in large animals, showing that we can insert these tissues, engineered tissues to large animals, pigs, spinal cord, and you can see by this MRI in the middle over there that there's a nice integration of the implant with the spinal cord. But sometimes we do not need to have a large tissue. Sometimes we only need small tissues, for example, for Parkinson's disease. And what we've decided to do here was to inject the hydrogel with the cells through oil to create tiny emulsions. Now we have these droplets. We can differentiate them, for example, to dopaminergic implants, as you can see here, and they secrete dopamine. We're now in animal studies and we hope to show soon that we can regenerate the Parkinsonian brain. We do the same thing with the cortical implants for brain trauma and stroke. And we can also print these cortical implants in arrays to recapitulate the gray and white matter and to study how these implants talk with each other from the cell bodies through the neurites and the axons and to the other implants. And you can see it here, how they interact with each other. You can see nicely the neurites. You can see it also by the FIB SEM images that can really dig into the implants, and you can see that these neuronal networks are real 3D. And when you look at basically at the function of these implants, you can see, I hope that you can see and appreciate how they interact with each other through the neurites that they grow towards each other. And so this is a model for us to study many diseases, brain diseases. So we're now back to the heart. I have a few more minutes. So I'll talk about the transfer of the electrical signal. Sometimes we're using materials that are not conducting. In this case, it was alginate scaffolds. You can see the pores. The cardiac cells are inserted into the pores. They contract in the pores, but they're not really interacting with each other on the electrical level. And what we've decided to do, and this was a long time ago, was to incorporate gold nanowires into the walls of the alginate to allow the cells to interact with through these wires with the other cells. And here you can see by these two movies the difference between without nanowires and with the nanowires. Everything is contracting together. And you can see in this graph that the patient-specific material with the gold nanoparticles, and this is a natural material that becomes fully conducting. You can see it by this IV curve. It works also with the neurons. You can see this is on day 3. You can see the extensions in neurons that grow on gold nanoparticles that on the fibers, and you can see the long neuroids. And this is extremely important when we talk about regeneration of the brain or of the spinal cord. Other things that we can do with the gold nanoparticles, for example, in addition to the better electrical coupling, we use the gold nanoparticles to absorb reactive oxygen species that are formed or generated after heart attack or after injuries. And we can also use gold nanoparticles to image the patch coverage in comparison to the scar tissue. So here you can see the better contraction that we have with the particles. You can see that we can reduce reactive oxygen species in the animals after the ischemia reperfusion model. You can see that we can evaluate or see the scar tissue that is formed and how the injected tissue covers it, and we can add more, we can inject more if we wish. And essentially, we can improve heart function, as you can see here, by this elevated ejection fraction. If you remember, I mentioned that there are stitches to secure the patch to the heart, which is not ideal. And here we use the gold nanoparticles optical properties. We illuminated gold nanoparticles in the scaffolds, and they locally heated and they were soldered. The patches were soldered to a tissue. This was in a 30-second illumination, and you can see how this patch is stuck to the heart without the need for stitches. We can also use the gold nanorods in this case to release different factors. So these particles are used as antenna for electroactive polymers, releasing factors, in this case SDF1, and bringing in more new blood vessels, as you can see, into engineered tissues. Last series of project that I'm going to talk about is what we call cyborg tissues, the integration of electronics with engineered tissues. So here we create nanowired mesh devices, porous electronics, basically incorporated the electronics to the scaffolding material to have scaffolds with many nanosensors within. This was because we wanted to evaluate the function of the tissue to see whether the cells contract or not. When we culture cardiac cells on these scaffolds, this is just one electrode, but we had many electrodes like that. You can see that we can see on the computer the function of the tissue. We can add drugs, for example, noradrenaline, and we can see how this drug affects the tissue. So it was a decent publication for us, a nice patent, but it wasn't enough. In this case, we could only sense the function of the tissue, but if the cells were not working properly, we couldn't do anything. So next, what we decided to do is to add another feature or two features. In addition to the recording, to the sensing ability, we wanted to add stimulation ability and release of drugs. So for example, if the patient is sitting in his house, is not feeling well, the physician can log on to the computer, can sense the function of the heart and decide how he wants to remotely activate it. For example, if there are not enough beats per minute, type on the computer, provide electrical stimulation. If he senses that there is not enough oxygen, type on the computer, release pro-angiogenic factors to create new blood vessels, or type on the computer when it senses that there is inflammation to release anti-inflammatory drugs. And these are not things that I imagine. These are actual things that we did in this paper. And we actually don't need the physician because we can create a code and the cardiac patch will basically regulate its own function. Oops. We can create more flexible and stretchable electronics, integrate them with the engineered tissues. We can now release up to 6 drugs in parallel in the right location, wherever we want, whenever we want, which drug we want. We can reload drugs into biomaterials just by modifying the drug a little bit to recognize biomaterials, and then we can inject the drugs into the circulation. It will find its way into the biomaterial and be released over and over again. And in collaboration with John Rogers in Northwestern University, he creates this cool pop-up electronics, 3D electronics, so we can incorporate it with our patient-specific hydrogels to create these tissues with electronics in different levels, or sensors within different levels of the tissue. We can also create piezoelectric scaffolds for cardiac tissue engineering. So here what we've done, created these materials that when they are deformed or they contract, they start to convert the energy to electrical signal. And when we cultured induced pluripotent stem cells and differentiated them to cardiomyocytes, we could see immediately when they started to contract, we could see it on the computer. But the electronics that we used so far were electronics that were made in the clean room. Their mechanical properties were not ideal for dynamic organs. There was a mismatch in the mechanical properties. They were stiffer, and this would lead to immune response. So what we've decided to do is to formulate a new electrode solution so that we can print the tissue with the same and the electronics, which has the same mechanical properties together. You can see it here. And eventually you can maybe appreciate how soft this tissue is and how flexible it is. And basically we can do the same thing, record and stimulate this tissue. We can create degradable electronics. And you can see that after a while, these electronics are just degrading and they're not functional anymore. And last slide that I'm going to show is what we call the engineering of a whole bionic heart. We can now engineer the heart with electronics all over, on the septum, on the ventricle, on the atria, on the pacemaker cells, and we can provide recording and stimulation. And with that, I will end up, acknowledge my group members, collaborators, and funding. And I would like to say that in a few months, we're going to have a Nature conference on nanobiotechnology for precision medicine and tissue engineering in Tel Aviv. So save the date. And I'll be happy to answer any question that you may have.
Tal, thank you so much. This was really an amazing spectrum of like top-down engineering modalities to build tissues and also to actually give them functionality. So thanks so much for the seminar. Before we switch to questions, let me just announce that we will take a break now over the Thanksgiving holidays. So just to be on the lookout in December for continuing with the tissue talks. So there is no seminar next week. Let's start with, there are actually two questions that are addressing the electrical signals and their conduction in the heart, in the engineered heart structures. The first one comes from Bhushan Karbikar. How is the directionality of electrical signals achieved in a cardiac patch? For example, how do you ensure that electrical signals originate from a specific point and propagate in a defined direction to enable directional contractions? So how much similarity there is with the conduction patterns that are observed in Purkinje fibers?
So basically, we can control the directionality or the propagation of the electrical signal in different ways. We can create, for example, the aligned fibers, which elongate the cardiomyocytes, and their connexins are really nicely expressed between the cells, and this allows to have a nice propagation of the electrical signal. This is with synthetic materials or with electrospan fibers. If we talk about the hydrogel, the patient-specific hydrogel, we simply put the cells within, and we allow the cells to remodel the materials to have more align fibers. We can also control the directionality of the fibers by the printing with the shear stress of the needle. This is another thing that we can do, but the best thing is actually to print the pacemaker cells or Purkinje fibers so that the signal goes through these fibers and to the entire tissue.
Okay. And related question asked by Beiping Chang, can a cardiac patch be made to beat at the same rhythm as the host tissue after transplantation?
Yeah, so this is something that we do not need to pre-design the tissue to contract in a certain pace. We just place it on the heart, and once the cells are integrated with the host dictates
Yeah,
the signal pace. So there's no need to think about it. We just need to make sure that the cardiac patches contract nicely, and once they are integrated, there will be a nice talk between the healthy part and the patch.
Okay, great. So a question by François Chesnay about the vascular component of your whole heart that is implanted into mice. So does this heart have its own vasculature and microvasculature? And if so, does it anastomose, does the anastomose in the omentum forms fast enough to avoid hypoxia?
So yeah, we were able to print the large blood vessels within, but these large vessels, they were enough to nourish the tissue, and we simply connected the entrance to these vessels and the outlet to the artery, so it was fully nourished. And when we looked afterwards on the histology, we could see that the cells are viable, and you could see that they contract nicely after we extract them from the body. So this means that they're viable.
Okay, okay. So question by Margaret Cruz. It is about actually built-in electronics. So is there a threshold for a long-term degradation or leakage of conductive nanowires or metallic elements like your gold particles in your patches? And if you have checked for any toxicity that is associated with this approach?
So yeah, first, there's no toxicity to gold in the body, not to nanowires, not to nanoparticles, nanorods, whatever. But of course, we checked it. We thought it's biocompatible, and even after the scaffolds are degrading, we couldn't find any
Any
toxicity.
toxicity. Okay. So I meant to ask you, like at one point you demonstrated that actually you can, with great precision, print different subpopulations of the cells into the heart structure. I was wondering, how do you see this like imposed, like a top-down approach where you are engineering the heart structure versus the subsequent process of these cells responding to the environmental signal? And if printing a little bit more immature structure may be beneficial for response to the heart environment and integration, both structural and electrical.
So we're still looking at it. We're still trying to understand what would be the best structure for the heart. I'll give you an example. Initially, we started by having the atria and the ventricles really connected, and the signal transferred through the atria to the ventricles. And then we understood that the ventricles and the atria should work separately. So now we're printing with a little bit differently, with just acellular material in between to be able to isolate these two chambers. So we're still trying to optimize the best way to engineer these tissues and how to mature them later on, which is even more important than the printing. The maturation is is critical. And actually, this is the limit now. This is our limit now, or the biggest challenge, how to, after we print it and after the cells contract, how we continue to mature so that it will the ejection fraction will be above 16%, which is not enough for now.
Okay. And maybe one more question, Tal. I mean, very clearly, what you do, the different kinds of heart structures and substructures have, like, there are two areas of utility. I mean, one is that these tissues may provide high fidelity models for answering some fundamental scientific questions, but then at the same time, you show that many of them really have translational value. So you are really looking into application in patients, and you are already being involved in clinical studies. So I wonder if you can tell us about the challenges in the first area and the second area. Where do you see the greatest difficulties, to come to the finish line, towards either like doing meaningful scientific studies versus translating into benefit to patients?
Yeah, so when we talk about regenerative medicine and we talk about simple models, not the heart, but the cardiac patches, we're now working actually on December 7th. We're going to start a big experiment in pigs with printed patches. And one of the challenges when we move on from rats to pigs and later on, hopefully, to humans, is how to create these really large structures. We need so many cells. If we created for the rats, we used a few millions. Now we use for each cardiac patch about 300 million cells, just cardiomyocytes, and this is a challenge. We have thicker tissues which need to be better nourished, so we have to have more blood vessels. So when we scale it up, and of course when we'll move on from a rat size heart to a full heart or a human heart, we'll have issues of scaling it up, more cells, more blood vessels, other ways to mature these hearts. It's still very far away, so there are so many challenges. And in terms of drug screening, for example, when we move on from these organ on a chip models to larger tissues, so now it's not a real high throughput. It's harder to create many of these tissues that repeats. So this is another challenge: how to create these cardiac tissues with the blood vessels, and to evaluate how each time the drug penetrates or goes out of the blood vessels in the same way, and the vessels form in the same way. So it's not like really thin layers of tissues. So there are still many challenges for each aspect.
Okay, Tal. So let me just thank you one more time on behalf of all of us, and thanks the attendees for really, really nice, insightful questions, and see you in a couple of weeks.
Thank you. Bye bye.
Bye.